warm welcome for Dr. Harvey Fishman.
Thank you so much for inviting me back, Carrie, and the organizers. This is a fantastic, I just love this conference. The people here are just terrific, and I just absolutely enjoy being here and interacting with everybody. Any case, so what I want to start off by saying is, what I'm going to talk about today is how the microbiome powerfully modulates our ocular health. And I'll get into that in a bit. I'm just absolutely passionate about the gut as an ophthalmologist, as a physician, and as a human being. So what is the microbiome? And this is something that will surprise everybody because you feel like every single TED Talk that you ever hear has microbiome. Somebody will talk about the microbiome. There are more TED Talks about the microbiome than there are bacteria in our colon. I mean, it's just remarkable. Now, what is the microbiome? Interestingly enough, Joshua Lederberg, who won the Nobel Prize back in the 50s, and he's kind of the grandfather. He really started this field of microbiology, or really did start it, but he was one of the big leaders in it. He actually coined the term microbiome in 2001. And he basically coined it, and he said, look, this is a term that to signify the ecological community of commensal, symbiotic, and pathogenic microorganisms that live all around us and all around our bodies. And so when you think about the microbiome, think about, hey, this was only a term that was coined in 2001, which I guess is, 17 years ago? It doesn't seem that long ago to me. 2001 seems like yesterday. So this is really cool. Genetic techniques for sequencing have really come a long way. And basically what this slide shows is that there are a bunch of companies now, and the technologies, as I said, of these companies were stuff that we worked on back in the early 90s. And basically they can take a single microbead or a single chip, and a little pixel on a little chip, and they can put a segment of DNA which is specific which is the 16S rRNA DNA segment. They can put it on one of those little chips. So like each one of these little pixels or a bead here becomes a single segment or a single type of DNA. And then you basically can take your bacteria and you kind of process it and chop it up, and then you place it on the chip, and when it binds, one of those things lights up. So you can be very specific about which DNA you have on there. And then they do this with these chips. Now this is actually a this is a microarray, a microfluidic array, where they can flow different DNA and they can do different processing. And what's really cool about this is this actually is what I did when I was doing my PhD. We were doing microfluidic chips and measuring small molecules, and we were doing things like neurotransmitter detection out of synapses and so forth. But using this technology, the big picture here is that prior, and I remember this distinctly, when I was a grad student, they were just starting to work on the Human Genome Project. And basically, and I was a grad student in the 90s, but in 2000, they finally were able to sequence the human genome. It cost $100 million. Think about that, $100 million to sequence the genome, okay? That was 2001. Now, what's really cool is a couple years later, it cost, and this was as recent as 2008, $300,000. Today, under $1,000. And probably in about 5-10 years, it could be like $5, $10 to basically sequence the whole genome. That is why the human microbiome and what we know about the gut is accelerating at such rapid pace, because it's just cost. It's a cost and it's a throughput device. And you can actually see here, this is a plot showing the cost per sequenced genome. It really went so quickly. This is like an inflection point. Back and gosh, I think I need glasses here. Can somebody help me? 2010, and it drops off, and then finally you're down to under $1,000. And this really is why we're where we are with understanding microbiome. This is a paper that shows kind of the where microbiome research is right now. There was a total of about 17,000 papers written on the microbiome, and this was a 2016 publication, but 10,000 of those papers were written in the last 7-8 years, okay? But what's really interesting is look where the eye is. there's only a few, maybe less than 100, couple hundred papers written on the eye and ocular microbiome. And so this is a really interesting area that has a lot of research potential, and where you're really going to start seeing an explosion in understanding eye health, and we're just getting into that. And part of the challenge of understanding the microbiome and the bacteria of the eye really turns out to be detection. So it goes back to my old wheelhouse, and that's sort of how I got interested in this, is because what I realized is that the way we culture, and on those beautiful papers that you did, the work that you did on, John, that you did with the culturing of the adenovirus. I mean, culturing is a sloppy technique, and it's imprecise. There's a lot of issues in terms of the right sample and so forth. And so one of my interests is being able to do microanalytical detection of bacteria, genetic genes, different types of mRNA, and other types of things on the surface of the eye. And that's really how I got kind of really interested in this. And that's one of the things that I'm working on right now.